This is my water quality lab report presentation by Reagan Souls, and it is in the Introduction to Science Class 101. To start off my presentation, I want to give a brief introduction about what the water quality lab experiment was about and about my wetland that I tested the water from. So in my water quality lab experiment, I tested and sampled water from a wetland that surrounds agricultural land. So to answer the question that was given to us about what is a wetland, it states wetlands are areas where water covers the soil or is present either at or near the surface of the soil all year or for varying periods of time during the year, including during the growing season. And that was quoted from the Environmental Protection Agency. And the wetland that I tested and sampled surrounds agricultural land, meaning surface runoff from fertilizers and chemicals could affect the quality of the water that I tested. According to the Environmental Protection Agency, pollutants in groundwater and fresh surface waters that flow into wetlands can be toxic to plants and animals, and they can accumulate in wetland sediments. And knowing this information regarding the conditions of the wetland before conducting my samples, this led me to some predictions regarding the conditions of my chosen wetland. When predicting my expectations of the condition of the wetland, I predicted that the wetland would be polluted, considering it is surrounding agricultural land. My predictions included I predicted that the dissolved oxygen was 2 ppm, and that it would mean it was very poor and wouldn't support aquatic life. And as far as my knowledge, there's no aquatic life present in the water source, so I predicted that the dissolved oxygen would be low. For the nitrate levels, I predicted that the nitrate would be very poor because there is surface runoff present, and since the water source surrounds agricultural land. And for the pH levels, I predicted that the water source will be slightly acidic because it is a fresh water source. Another quote that I found was, runoff from agricultural land and even our own yards can carry excess nutrients such as nitrogen and phosphorus into streams, lakes, and groundwater supplies. These excess nutrients have the potential to degrade water quality, and I found that from Water Science School. And in the journal article, Advancing Surface Water Pesticide Exposure Assessments for Ecosystem Protection, the authors state, agricultural systems involving pesticide use and management are complex and are located within natural landscapes where different and often unknown components interact. These systems are very difficult to predict because interactions within the system often result in large uncertainty and lead to emergent behaviors, giving rise to a complex food, energy, water, social nexus problem. The authors further explain why the use of pesticides should be managed, stating, management of source water quality and pesticide use will be important to support sustainable food production systems and environmental quality. And that all came from Fox et al. And to finish up my introduction, my hypothesis states the water at the site is not healthy. This hypothesis was made based on the existing knowledge of the water source surrounding agricultural land and the possibility of surface runoff from fertilizers and chemicals being present. The purpose of my research and experiment was to determine the water quality and the health of the wetland after multiple samples were taken and tested. The method I used consisted of taking a sample of water and placing it into a Tupperware container. Right away, I took the temperature because I wanted it to be as accurate as possible. And once I was home, I tested the dissolved oxygen using 2 tablets in the small glass vial. And I tested the nitrate using 2 teaspoons of water and placing one of the nitrate tablets into the small container. And I tested the pH using the same small container, fitting 2 more teaspoons of water with 1 pH tablet. And lastly, all samples and results were recorded in the water quality field data sheet. So the materials that you need for this experiment are 12 dissolved oxygen tablets. You need 6 pH tablets, 6 nitrogen tablets, 1 small glass vial for testing the dissolved oxygen. You need 1 water collecting container. I just kind of use a circular Tupperware container. You need 1 teaspoon. That can either be just like a teaspoon, like a regular spoon, or you can use your teaspoon measuring tools. And you need 1 small clear container that you can fit the 2 teaspoons in. And you also need 1 thermometer to test the temperature of your water. So for my method, the wetland that I tested the water quality from is a water source that surrounds agricultural farmland, like I said previously. There's lots of plant life and organisms that surround my wetland, making it full of biodiversity. Whenever I went to test the water, I saw multiple organisms, such as I saw a female deer run out in front of me. I saw multiple species of birds, multiple species of grasshoppers, crickets, butterflies, frogs. Anything you can think of, really, I saw in like a farm environment. And the water source itself is scarce, as over the many years of agricultural farming and increasing temperatures, especially in the summer here in South Carolina, they have both had a great effect on the wetland that I tested. So a more in-depth paragraph of my method. When determining the water quality of my wetland, I sampled and tested the water 6 times. The sampling occurred on the dates of July 12th, July 14th, July 17th, July 19th, July 22nd, and July 25th. When sampling and testing the water, I first filled my bucket with water from my wetland. I then took the temperature of the water using my thermometer right away to get the most accurate results. When testing the dissolved oxygen level of the water, I submerged a small glass vial provided into the water sample. I then dropped 2 dissolved oxygen test tabs into the tube. I screwed the cap on the tube and made sure that no air bubbles were present in the sample. I mixed the sample by inverting the tube over and over until the tablets had disintegrated. I waited 5 minutes for the color to develop and compared the color of the sample to the dissolved oxygen color chart. Lastly, I recorded the result as ppm dissolved oxygen. When testing the nitrate level, I used a small container and filled it with 2 teaspoons of water from my sample. I then added 1 nitrate test tab, covered, and mixed until the tablet dissolved. I moved the container into a dark area, although the instructions said that as long as you were inside, it didn't matter, but I wanted to get the most accurate results, so I did place it in a kind of dark area in my house. So I placed it into a dark area, and I mixed the sample for 2 minutes to disintegrate the tablet and waited 5 minutes for the color to develop. I then removed the tube from the darkness and compared the color of the sample to the nitrate color chart. Lastly, I recorded the results as ppm nitrate. Finally, when testing the pH level of the water, I cleaned my small container completely by rinsing thoroughly with regular water after the nitrate was complete. I filled the small container again with 2 teaspoons of water from my wetland and added 1 pH wide range test tab. I mixed the sample until the tablet was disintegrated, and lastly, I compared the color of the sample to the pH color chart and recorded the results as the pH. These are just some pictures of my sampling. So this one in the top left is just the materials that I used. This one right below it is me testing the temperature of the water. This one right here is just one of the results of one of my dissolved oxygen samples. This one below it is the result of one of my pH levels. This one to the right is the results of the nitrate levels. This one right here below that one is me pouring 1 teaspoon of water into one of the small containers. I can't exactly remember which one I was testing. I was either testing the nitrate or the pH. This one in the top right corner, just another picture of my materials that I used. And this one in the bottom right corner is me dropping one of the dissolved oxygen tablets into the small glass vial. So now on to the results of my experiment. So after sampling and testing my water over the 3-week period from July 12th through July 25th, my sampling results are as follows. So my pH levels kind of maintained a pretty consistent rate. They were either 7 or 8. My dissolved oxygen levels were 4 ppm through the whole experiment. My nitrogen levels were 5 ppm throughout the whole experiment. And the temperature of the water ranged from 24.44 degrees Celsius to 26.67 degrees Celsius. And I also provided my water quality data right here. And just to the right, you can just see like it also asks for the weather. So the weather was pretty consistent. The first day it was overcast or cloudy. The next 3 days were sunny. The 5th day of sampling was cloudy, and the 6th day of sampling was just partly cloudy. So on to my results. The pH levels of my water source was the only aspect of my experiment that had any amount of change, with my dissolved oxygen levels staying constant at 4 ppm and my nitrogen levels staying constant at 5 ppm. The pH levels, the pH was the most significant amount of change that I saw in my experiment, other than the temperature of the water. To present and demonstrate this amount of change in the pH levels, I have provided a line graph of the pH, bar graph of the pH level change throughout the course of the experiment and on each day tested. And I also provided a bar graph of the dissolved oxygen levels and the nitrogen levels. And onto more of my results. I found a potential relationship between the pH levels and the temperature, so I'll just explain that. When observing my data and results of my experiment, I observed a potential relationship between the pH levels of the water with the temperature of the water. There is a similar trend between the two sets of data, but they are not exact. I observed a trend of the pH level being 7 around the temperature of 25 degrees Celsius. So, similarly, the temperature, whenever I saw a pH of 7, the temperature was either 25 degrees Celsius on the dot or around 25 degrees Celsius. The pH was not at a level of 7 every time when the temperature was around 25 degrees Celsius, but that was a trend that I observed when looking closely to my data and results. And I provided a line graph of the trends of the temperature and the pH level. And you can see, I'll put it a little bit closer. You can see the trend is not exact, but there is some similarities with it. So that's why I wanted to include that potential relationship. Moving on. Now on to my discussion. The testing and sampling of my water site resulted in a pH of 7 to 8, dissolved oxygen levels of 4 ppm, and nitrogen levels of 5 ppm. When comparing my sampling and testing results to the ranking results provided in the sampling procedures, my water source was not completely unhealthy like I thought it would be. So, as my hypothesis stated, I did believe that the water site would be unhealthy, but according to my results, my water source was actually pretty healthy. According to the pH ranking results, with my levels being between 7 and 8, a pH of 7 is considered excellent and the pH of 8 is considered good. The nitrate ranking results conclude that my levels of 5 ppm are fair in a water source, and with my dissolved oxygen levels being at 4 ppm, this leaves my water source being in between stressful for aquatic life and at a good level. So it was kind of in between. There isn't really any aquatic life that lives in the water, but there is some organisms that surround it, such as plant life and other like tadpoles, obviously, just stuff like that live in the water. I would not consider my hypothesis being rejected according to the results that I obtained, but I would also not consider my hypothesis as being accepted. So my hypothesis, like I said before, I did predict that the water site would be unhealthy. So from my results, it is kind of considered unhealthy, but I wouldn't say that it is completely unhealthy. So that is why I would not say that my hypothesis is rejected, but I also would not say that it is accepted. I believe that more sampling and testing of the water site needs to occur before the water source can be determined as healthy or unhealthy. Leading into more of my discussion, as stated previously, my hypothesis was neither rejected nor supported. Before performing my experiment and testing the samples taken from my water source, it was assumed that the water would be unhealthy considering the agricultural land that it surrounds and the fertilizers and chemicals that would possibly be present in surface runoff. Considering the certain fact that the area is agricultural land and crops are grown and produced year-round, the water source must not be close enough to the agricultural land to be affected by surface runoff. The area of where my sampling took place was a considerable distance away from the farmland, so this is why the water source was not completely unhealthy. Referring to the background information that I provided in my introduction, the Environmental Protection Agency states, pollutants in groundwater and fresh surface waters that flow into wetlands can be toxic to plants and animals, and they can accumulate in wetland sediments. And this was from the Environmental Protection Agency. No plants and animals were present in the actual water source, so the fertilizers and chemicals present near the water source have to have some effect as to why no organisms were present. My water source did not have the greatest amount of water present, so finding a source in the same vicinity with more water would help to improve my experiment when furthering my research regarding my water source. Now on to my conclusion. When performing my experiment, it consisted of gathering samples from my wetland that surrounded agricultural land, then testing my samples to determine whether my water source was considered healthy or unhealthy. When sampling and testing, I tested for dissolved oxygen levels, nitrogen levels, and pH levels. The results of my testing provided me with the information that my wetland was not completely unhealthy like I assumed and predicted. The dissolved oxygen levels were in between stressful for organisms and good, so it was in between. And nitrogen levels were fair, and pH levels were good and excellent at certain times. When determining my major findings of my experiment, it can be concluded that surface runoff resulted from fertilizers and chemicals do have an effect on the water source that surround the area. And I believe that if no agricultural production of crops were present around my wetland, it would be able to provide a habitat for organisms and the environment would be healthy overall. But this cannot be officially concluded unless further and more extensive research was carried out on the water source in the wetland. And I would just provide a little background. I've been living in this neighborhood practically my entire life. So I have grown up around this farm that I got my water source from. And I can say that years and years ago, there were some organisms present in the water source. Like there used to be lots of beavers that would build dams. Fish were in there, just stuff like that. So I can say that organisms were present, but I really do believe that with all of the agricultural production that has occurred on the farm, that it has slowly but surely made an effect on the water source, and the water source has drastically declined. And I think that the agricultural production has had a factor in that. And here is my bibliography. I got some of my background information from the article Advancing Surface Water Pesticide Exposure Assessments for Ecosystem Protection. I got some more of my background research from the article Runoff, Surface and Overland Water Runoff, and that was by Water Science School. I also got some of my background information from the article titled Wetland by the Environmental Protection Agency. And I also got some background information and the answer of my question, what is a wetland, from the article titled What is a Wetland from the Environmental Protection Agency. And that is my water quality lab experiment. Thank you.